Hello, last section on Ebola virus. Here we go. And here's an Ebola virus particle. This is the famous early image. And this is that sort of end of it that's curled around. That was the first picture that everybody saw of Ebola, and so they thought, oh, Ebola always looks like this, and it doesn't. It usually looks like just the top part here, like a long stretched out sausage. Okay, so this ends up being essentially immunology, but it's viral immunology. And that's a real important component of virology because that tells you both why you get sick and how you get better. So, all right, if you've had immunology, there's going to be some review here. And if you haven't, you know what? Here's a little baby immunology course for you. And yeah, all right, let's get into it. Let's get into it. All right. So, first of all, you've got your infected cells. And so these guys are going to release interferon alpha and beta. And so it'll probably be those, like those endothelial cells. Here we're talking about just a general sort of infection rather than Ebola infection. But in the case of Ebola infection, remember, it doesn't really work on anything except the lining of your veins and arteries and some of those cells of the immune system and cells in a couple of your major organs. So infected cells will release type 1 interferon, which is going to be interferon alpha and beta mostly. And that's going to signal to start antiviral responses. We know about that. That's good. Then let's see, dendritic cells are going to be out here, and immature dendritic cells are going to be attracted by the interferon, and they're going to capture antigens. So they're just going to wander over and, like a bored security guard, see what's up, see why we're putting out interferon, and see if we need to radio on the old walkie-talkie to mall headquarters. And in this case, what the dendritic cells will also do is they'll pick up a little sample of whatever it is that's happening there and try to take this to a lymphoid organ, something like a lymph node, or something more like the spleen, where they can show it to other smarter cells of the immune system. Yeah, the dendritic cell is not the sharpest tool in the box, but it is functional. It does its job. Yeah, very much mall cop. No offense if you are a mall cop. Yeah, okay. So dendritic cells can also release massive quantities of interferon alpha, which will bring in more dendritic cells, which will cause more little pieces of this to get carried back. Now, the immune response is going to depend on one dendritic cell that has picked up an actual piece of the virus, wandering into just the right T lymphocyte that just happens to have genetically recombined itself to a point where it can bind this. And then, boom, if they make a connection between those two cells, which there may only be a few, like 5 or 10 of each in the body, but if they find each other, then boom, the white blood cells will start to differentiate and divide, and you will make a great immune response. But there's a lot of chance there. There are a lot of lymph nodes that these dendritic cells could go to. There are a lot of different places where T lymphocytes hang out, and every one is genetically a little different from all the others. And so, yeah, it's like a lot of luck, like an unacceptable amount of luck behind any immune response. And so a dendritic cell's job is to do what we're seeing down here. This is a little KISS, the interface between the dendritic cell, which is this spiky dude over here, and the lymphocytes. These are a little T lymphocyte. And the dendritic cell is going to actually pass over that little piece of Ebola virus to the T lymphocyte, and if it's able to bind it, then the dendritic cell is going to say, well done, and the T cells, lymphocytes are all going to say, yeah, you did it. And they're going to do that with chemicals and cytokines, of course, rather than with little cheers or emojis. But that is going to make the T lymphocytes start to differentiate, and that's going to make it start to, we call it expansion, where it just divides and makes more copies of itself. So dendritic cells can also present antigens directly to B and T cells. And so dendritic cells can directly cause other cells to produce antibodies or make a killer T cell response, which is very cool. They can also present to helper T cells, and then helper T cells can go out and do the same thing. So they're like a smart version of a dendritic cell that can help a lot of other T cells or B cells. Um, but Ebola virus, yeah, will bind to DC sign on the dendritic cells. So remember, the DC part stands for dendritic cell, and the sign part, oh, I forget what it stands for, but it's something convoluted so that we could just say that, oh, this protein you find on dendritic cells is the sign of dendritic cells. DC sign, yeah. Um, yeah, the virus will actually hitch a ride to the lymph node where it's going to run into some macrophages and infect them and cause a terrible outbreak. So, the way it should be working is the dendritic cell picks up pieces of Ebola and brings them to show the T cells, and boom, we get an attack, and the interferon sort of marks where we should attack. What happens in Ebola is that the dendritic cell picks up an intact Ebola virus, tries to show it to other parts of the immune system, but ends up giving the entire infection to macrophages, which then lose all of their compounds, all of their cytokines and chemokines, cause that cytokine storm, and eventually kill the host, which is you. Yeah, so that's bad. All right, back to genetics, because why the heck not? Yeah, genetics play a big role in every infection, determining who lives and who dies. We have a really good, clean story that we can tell. We have a decent understanding with Ebola virus, and we don't have that same understanding for some other viruses. And so that's why I picked Ebola virus as the one where we tell this story. But there's going to be some similar story. There'll be certain genetic versions, genetically inherited versions of major histocompatibility complex alleles that will absolutely protect you or kill you in different kinds of infection. And yeah, that's just genetics and the luck of the draw, right? So remember, we were talking about human leukocyte antigens, HLA, and HLA types A, B, and C are all together called MHC class 1. Together they are the major histocompatibility complex. And so HLA-A and B and C are all made on normal cells, and they're all going to be found on chromosome number 6 right here. And HLA, DR, DQ, and DP are going to be found. These are things that are going to be made in T cells, so in cells of the immune system, let's just say. And so don't worry about those. But those are very important for tissue transplants. But yeah, if you look at it, there are actually quite a lot of these. So there are a ton of different HLA-As, 309 that we know of. But look at the HLA-Bs, 563 different types scattered among humanity. So at most, you're going to get 2 of these, one from mom or one from dad. But if the luck of the draw says that your mom and dad just happen to have the same one out of 563, and so it would happen, then you basically only got one. You got one to carry you through life. And yeah, hope it's a good one. Hope it really works out for you. All right, interesting little aside that doesn't really particularly matter, but why the heck not? So, major histocompatibility class is also involved not just in immune reactions, it's also involved in love. Yeah, that feeling that you think you feel, yeah, on Valentine's Day or whatever, yeah. Partially driven by MHC class 1. Here's how. So this has been shown in mice and actually quite a number of animals. There are some results that suggest it's probably happening in humans, but to a lesser extent. But the idea is that female mice, so male mice will basically mate with anything, but female mice exhibit a mating preference. They won't let just any male mouse mate. They choose. So if you put them in a cage with several male mice, they will mate with one, but not the others. And it'll be consistent. And so when the female is ready to mate, she is actually sensitive to genetic differences. And she can basically smell genetic differences and will attempt to mate with a male that is genetically more different from her, or at least has a different MHC class 1 molecule, which, you know what, has the effect of ensuring that the kids are going to end up with 2 different ones, right? Yeah. And she will be repelled by mice that have the same exact MHC class 1. And since mice, lab mice tend to be clones of each other, you can test these things out and show that it happens over and over. So on the surface of cells in the nose, there is MHC class 1. And those MHC class 1s hold on to little tiny peptides, little piece of whatever that cell is making at the moment. So it doesn't matter if it's viral proteins or cellular proteins, whatever the cell's making, we're going to chop up a little part of that and lock it onto MHC class 1 and show the world what we've got. So when we do this in the bloodstream, that's fine. White blood cells can see it, and they can know whether or not that cell is making something it should not be making and whether to make an immune response. But these proteins are also expressed on the outside of the nose, the side that comes in contact with the air. And so when you breathe out, you're actually going to breathe out. Some of these little peptides will come loose and they'll flow out in the air from your nose. And when that happens and somebody's next to you, those little peptides can go into that other special someone's nose. And then if they dock onto that person's MHC class 1, that means you two probably have either the same MHC class 1 or a very similar MHC class 1, and that will be perceived as revulsion. So if you smell somebody and there's just something off, just something like, oh no, yeah, where that probably comes from, and where that definitely comes from in mice, is a genetic match. The peptide from one has matched onto the other. So yeah, think about that next Valentine's Day. And but I guess you can look at it this way: if you find somebody who you like the smell of, they're probably not going to be a great organ donor. But if you find somebody who you can't stand the smell of, chances are they may have the same MHC as you, and so maybe you can get a kidney someday. I don't know. Is that worth it? Is that payoff worth it? Who can tell? You decide. Of course. All right, so here's the top of an HLA molecule. And you can see it looks like one of those binder clips. So you got an alpha helix here, and you got kind of a weird bent alpha helix there. And pinched between those two, these little blue things, are lots of different peptides. So they've modeled this with lots of different peptides, and the peptides always sit in the same spot. And so this is just, doesn't matter what peptide it binds, it's going to be clamped right between those two little alpha helices. And so this is the top, and that's the bit that's going to be presented to the rest of the immune system. And so that part is going to be right up here. Here's that little alpha helix that's going to present. So that's the same thing up here. And this is the rest of the molecule. This is the intact molecule modeled out. And so we're only looking at the very tip top. There's this little domain up here at the top. But that's the part that binds the peptide. That's the part that does the work. And so depending on exactly which version of the MHC class 1 or human leukocyte antigen, remember they mean the same thing in people, depending which one you inherit, you can only bind certain peptides. Each one binds to different peptides because each one has genetic differences all along this helix and all along this back helix. And those peptides determine what little bits of protein the HLA molecule can bind to. So this is how it goes. Let's say let's say you've got an antigen-presenting cell, like a macrophage or something like that. And let's say it gets directly infected. That's okay, because the antigen-presenting cell chops up a little piece of itself, puts it out via MHC class 1 or class 2, depending on who is trying to show it to. And you will then either get helper T cells recognizing it and calling for more help from killer T cells, or you can get killer T cells directly recognizing that little viral peptide. And yeah, they'll sort of clamp down on it, and then they will perforate this cell with perforin and kill it. The other way it can work is that you can have any sort of virus-infected cell that can, so it's going to present a little peptide on its surface to a dendritic cell. The dendritic cell has MHC class 1, is going to take that peptide and bring it over to the immune system in a lymph node. And there is our MHC class 1, and there's our little peptide, the little green thing on top. And so it'll again show it directly to a CD8 killer T cell. And if that killer T cell doesn't like what it sees, then bam, it will kill the dendritic cell, and it will make more copies of itself that will hunt down the original cell that had the virus and go off and kill it. And this whole process happens over the course of a couple hours once we've finally made the connection. But it can take a couple days to actually make this connection and find just the right T cell that's able to recognize this as foreign. All right. So there are many different kinds of HLA type B. Remember, there were 500 and some different types? And for some of these, we know exactly what it is that they are looking for in a peptide. Some other ones are so rare that some people have studied them, but there may be some that haven't even been studied. Then if you get one of those, you're just a genetic freak. And what can you do about that? Nothing. So HLA-B7, you can see over here is in this one, along with HLA-B51.02 and HLA-B35 version 01, etc. And all these guys are looking for proline amino acid at the second position. And so if you look, there are only certain ways that a protein can be cut, because there's certain machinery inside the cell called proteasomes that cut the proteins, and they'll only cut at certain structures and at certain amino acids. And so there's only so many ways you can chop, slice and dice an Ebola virus. And of those ways, there's only so many that would have a proline at the second amino acid. And so this is a comprehensive list of everything that a cell could make from all the Ebola proteins load onto an HLA type B7. You can see some of these other ones down here. B27 is going to look for arginine at the second position, which is fine. Sometimes you'll have peptides that look like that. Sometimes you'll have peptides with a hydrophobic on the 9th position, Sometimes you'll have a magic peptide that binds both of these things. That's what B7 is looking for. And so different HLA-B types are looking for different peptides. And the peptides are constantly being generated by every cell. And so that's what they're presenting. That's what they're showing to the outside world. And remember that HLA-Bs are just one of the types of MHC class 1. So if you wanted to sequence your own, it's actually not that hard. And this is what they would do to find out if you're a good tissue match. You can order the little primers from this particular paper, and you can read it out. You can make these little PCR products with those 2 primer sets. And you can send them off for sequencing. And the sequencing is about $5, and the primers are about $5, and maybe $1 worth of enzymes. So around $15 total to run this thing and figure out some of your own genes. And then you'll get out a thing that looks like this. So, for example, this would be one person's full HLA type. So for HLA-A, so this is MHC class 1, the first version, this person has HLA-A1, subtype 1, and 3 subtype 1. So one's from mom, one's from dad, and we'd have to sequence them to figure out which is which. And so then there's type C, there's your type B, so this is a B07 version 2 and a B08 version 1. And then these are the ones that are going to be expressed on T cells, DR, DQ, and DR, DQA, and DQB. There we go. And here's our little chart again. So this is how they came up with this test. There was an Ebola outbreak. It had a total of, 77 patients, where they had blood samples on all of these patients. 35 of those lived, 42 of those died. And so it was a little worse than 50-50 ratio living, something like 60% fatality. But they managed to get HLA-B sequences for some of these people. And so that's how we built up the first understanding of which human leukocyte antigens, which HLA types are associated with good and bad outcomes in the case of Ebola. And so here's HLA-B7. And so you had 11 patients that had it, and they all survived. 0 died. And so remember, HLA-B7 binds peptides with proline at the second position. And if it can bind those peptides, and there are a whole bunch that Ebola can make, then it can show them to your immune system, and your immune system can make a good response, and you will survive. That's the idea. So, yeah, do you know your HLA genotype? I actually don't know mine, but I'm very interested to find out. So, there we go. HLA B7 led to survival, and some of the others, B15 or 67, led to death. So, for example, Britain and most of the USA would actually be very similar. This is the proportion. So 28%, so about a quarter of people, and this is throughout Europe and the Americas actually, would have HLA B7. It's a very common type. And so if you have this, you're going to be in that group where everybody who's had this genotype and was in that particular study survived. So it seems to be protective. I'm not guaranteeing that you can go, into a bat cave and rub yourself in bat feces covered in Ebola and survive. But yeah, I think you'll have a better chance than others. And these percentages are shifted in sub-Saharan Africa, which is the place where Ebola tends to be found. And unfortunately, there are people who are there who have actually a little bit higher likelihood of having some of the HLA-Bs that do not help against Ebola. Now, the reason for this is probably because those HLA-Bs are going to help out against something like malaria or one of the other diseases that's much more common down there. Because no HLA type is good against everything. I mean, proline on the second amino acid, that'll protect you sometimes, but it won't protect you every time. And sometimes you're going to need lysine on the third amino acid or something like that to make a response. Because every virus, every bacterium is different. And so it's just a matter of, did you happen to inherit the right machinery that can cut it into the right pieces and show those pieces to the right cells? And then once you have all that machinery, do those two right cells happen to meet quickly enough to have a good outcome in the infection? And that's it. That's the many levels where it kind of comes down to genetic luck, whether or not you live or die in an Ebola infection. I'll do a little chalkboard thing on this to help bring this home. But there are a lot of very good papers on this subject, including the ones that I've referenced that I got these statistics from. And if you're ever not sure, just type in a couple words from like one of the figure legends on there. I usually copy the figure legend out, and that'll usually lead you right to the paper, the original paper, which will help you in answering the essay questions. So, thank you very much. This has been Ebola, and I'll do one more little chalk talk, and then that's going to be it for Ebola.